My name is Brittany Roberts, and today's date is September 9th, 2021. Case 1. Okay. So, transamination is the process in which ammonia groups are transferred from an amino acid to a keto acid, or transferred from a keto acid to an amino acid. This reaction occurs with amino transferases. It requires a cofactor pyridoxal phosphate. Deamination is another form of ammonia transfer, and here are enzymes that are used in this transfer. Glutamate dehydrogenase requires NADPH or NADH, and is able to fix or free ammonia from glutamate, within the liver. Glutaminase is also within the liver, and it frees ammonia from glutamate. And glutamate synthetase fixes ammonia to glutamine that is found within the peripheral tissues. It's important to note that the ammonia freed from glutamate or glutaminase are used in My name is Brittany Roberts, and today's date is September 21st, 2021. Case 1. Okay. So, ammonia transfer can occur with transamination or deamination. So, transamination is the process in which ammonia groups are transferred from amino acids to keto acids, or they can be transferred from keto acids to amino acids. This process occurs using amino transferases, and it requires the cofactor pyridoxal phosphate. Deamination is the removal of amino groups or ammonia, which is the same thing from an amino acid. Here are 3 enzymes that are used in deamination, and it's important to note that the ammonia that is removed using these enzymes are used in the urea cycle. So, glutamate dehydrogenase requires NADH or NADPH, and it fixes or frees ammonia from glutamate. And this process occurs in the liver. Glutaminase is also found within the liver, and it frees ammonia from glutamate. And glutamate synthetase fixes ammonia to glutamine, and this is a process that occurs in the peripheral tissues. Okay, so nitrogen transport is connected to gluconeogenesis because transamination and deamination provide substrates that are used in gluconeogenesis. 2 of these examples are pyruvate and alpha-ketoglutarate. So, alanine is transaminated to pyruvate, and glutamate is deaminated to produce alpha-ketoglutarate, which are both used as substrates in the production of glucose during gluconeogenesis. Okay, so here are the enzymes that are used in the urea cycle. The first clue in determining the enzyme that is deficient is the normal or lower level limits of normal of the citrulline, of the plasma citrulline. This level means that the deficiency is not affecting citrulline synthesis. This cancels out a deficiency, and the deficiencies in the enzymes arginine synthetase and arginine succinate lyase. If there was a deficiency in either of these enzymes, citrulline levels would be elevated and higher than 55, because the synthesis of arginine succinate would be affected. Yes. And a deficiency in arginase is very unlikely because this enzyme is in the last part of the urea cycle, and elevated ammonia levels is highly unlikely because of this. And in addition, because the patient's ammonia levels were elevated after only 24 hours, that's another reason why it is highly unlikely that this is a deficient enzyme. This leaves carbamoyl phosphate synthetase and ornithine transcarboxylase. Carbamoyl phosphate synthetase 1 combines ammonia and carbon dioxide to produce carbamoyl phosphate, and ornithine transcarboxylase combines carbamoyl phosphate, which was produced in the previous pathway, with ornithine to create citrulline that is then shuttled out of the mitochondria. It is important to note that carbamoyl phosphate has an alternative pathway outside of the mitochondria. This pathway occurs when there is excess carbamoyl phosphate. So the alternate pathway is the synthesis of pyrimidines with the use of carbamoyl synthetase 1. And an intermediate of this synthesis is orotic acid. For this reason, the elevation of orotic acid means that carbamoyl phosphate synthesis is occurring. In addition, it means that there is an excess of carbamoyl phosphate. For this reason, we can assume that carbamoyl phosphate synthetase is not the deficient enzyme. So for this reason, this leaves ornithine transcarboxylase as the deficient enzyme. This is demonstrated by the excess carbamoyl phosphate that is needed to be used in the pathway for protein synthesis because it is unable to be combined with ornithine to create citrulline. Because it is clear that carbamoyl phosphate synthesis is occurring, it is highly unlikely. This is the least likely enzyme to be deficient. And if there is a deficiency in the urea cycle, arginine becomes an essential amino acid. Okay. For scenario 1, this is most likely a deficiency in the enzyme branched-chain alpha-keto acid dehydrogenase complex, which is known as the maple syrup urine disease. This disease affects the metabolism of branched-chain amino acids such as isoleucine, leucine, and valine. So the first step in the synthesis of these amino acids is the removal of the ammonia group from an amino acid, the transfer of an ammonia group from an amino acid to a keto acid. This is followed by oxidative decarboxylation using branched-chain alpha-keto dehydrogenase to produce intermediate cerulated using the TCA cycle. Due to this deficiency, as mentioned, isoleucine, leucine, and valine are not metabolized, and as a result, they build up within the body. There are no amino acids that become essential with this deficiency. In fact, restriction of proteins is a typical treatment due to the inability to be metabolized. It's the restriction of the branched chain amino acids are typical treatment because they cannot be metabolized. There are various types of maple syrup urine disease, and one of them is caused by a deficiency in thiamine. For this reason, thiamine supplements may be useful for treatment because it is a cofactor that is required for branched-chain alpha-keto acid dehydrogenase. This means that it must be present for the reaction to occur, and if it is unavailable, then it will not occur, which prevents protein catabolism. Scenario 2. So here I have a photo of tyrosine metabolism. In scenario 2, a deficiency in the enzyme homogeneous 1,2-deoxygenase is also known as the disease alkaptonuria. This disease is important in the pathway of tyrosine metabolism. This particular enzyme is responsible for breaking down homogentisic acid into this acid right here. As a result of this deficiency, tyrosine is unable to be fully metabolized, and there is a buildup of homogentisic acid. This buildup can occur within connective tissues, both intracellular and extracellular, as well as within organs. The history of aortic stenosis and the pigmented macules demonstrate the deficiency of homogentisic 1,2-deoxygenase, because these are both examples of clinical representations of a buildup of homogentisic acid. There is no amino acid that becomes essential due to the deficiency. Once again, typical treatments for this disorder is the restriction of proteins because their metabolism is affected. Vitamin C supplementation may be beneficial for treatment because it slows down the oxidation of homogentisic acid to benzoquine acetate. Benzoquine acetate, as mentioned earlier, is the cause of the pigmented macules. So although it would not improve tyrosine metabolism, it would reduce some of the side effects that are experienced due to the deficiency. So in scenario 3. In scenario 3, there is likely a deficiency in phenylene hydroxylase, which is known as phenylketonuria. This is demonstrated by elevated levels of phenylalanine in the urine. Phenylalanine hydroxylase is an enzyme that is used to convert phenylalanine to tyrosine. Therefore, a deficiency in this enzyme would mean that phenylalanine is no longer metabolized. As a result of this deficiency, tyrosine becomes an essential amino acid because it is no longer created or synthesized in phenylalanine metabolism. BH4 is a cofactor that is required for this reaction and may be useful in treatment of the disease because this vitamin must be present for the reaction to occur. So if there is a deficiency in that enzyme, it will not occur, or it could improve the reaction.